Hello, everyone. Thank you so much for joining our e-seminar series on Translational Biomedical Engineering. Today, it's our great pleasure to host Professor Gervais from Polytechnique of Montreal. And let me share my screen. Okay, hopefully you will see the slides. Mohsen, just let me know if you see the slides. Okay, Professor Gervais will talk about theory-guided innovation in microfluidic from 3D culture to advanced reagent delivery. This is our 94th seminar from the beginning of the pandemic, and we will hear about his journey towards. Yes.
I can't see the slides. I don't know if that's the case for everyone else.
Okay. Nobody can see it. Let me just share it. Okay. So do you see my screen?
I can see your screen.
Mohsen, do you see?
Yeah.
Okay. So we will hear about his journey towards from bench to commercialization, from bench sites in the lab to commercialization. So what I want to say that before going to his presentation, we have a chat box. So the audience are encouraged to put their affiliation, their expertise, and where they are coming from, and talk to each other, and also like exchange ideas and kind of expertise to each other. And there's a Q&A box on the bottom of the screen that you can ask your question from our speaker. Next week we are hosting Professor Catherine Granfield from McMaster University. This talk is also sponsored by Canadian Biomaterials Society. For getting the more updated information about the upcoming seminar series, you can always look at the Twitter. Our handle is TransLBME, or our co-organizer, myself and Mohsen, also we post on our LinkedIn, so you can get the information about the upcoming seminar series. So this would not be possible without the help and sponsor from Montreal TransMedTech Institute, which has been with us from almost the beginning of this e-seminar series. By that, I would introduce our speaker very shortly. Professor Gervais is a professor of engineering physics and biomedical engineering at Polytechnique of Montreal and researcher at Centre Hospitalier de l'Université de Montréal, CHUM. He's also a member of Montreal Cancer Institute. His research deals with fluid physics and transport phenomena at the microscale and development of lab-on-a-chip strategies to accelerate discovery or improve treatment in oncology. He's co-founder of the CHUM's microfluidic core facility and of MesoChip company, a laboratory spin-off seeking to develop microfluidic tools to culture and analyze primary tumors. He holds a bachelor's degree from Polytechnique Montreal in engineering physics and a PhD in bioengineering from MIT. By that, I would invite him to share his screen and start his talk. The floor is yours. The virtual floor is yours.
Thank you very much, Professor Savoji. So let me try to share here. So can everyone see my presentation full screen now?
Yes, that's perfect.
Very good. So thanks a lot for having me. Just before I would, while I do not see the chat box, I would like to encourage you and Mohsen to forward me any questions live. There's no problem in interrupting me. Sometimes it's easier to answer them in context. And I would also very much like that the audience type who they are and where they're from in the chat box, so that I can have an idea of who I am talking to. This is very stimulating for speakers as in virtual mode, as you know. And then I can also gear my presentation slightly more towards the majority of the audience. So this is going to be a microfluidics for translational science talk, but there's it's science loaded. So there's a part of engineering design and also transport theory in this. And what I would like to do today is to showcase a little bit the three major fields of innovation in my research, which I will describe quickly. And the idea from my laboratory, I would not say a theoretician, but I would say I have inclinations toward theory, even though we like very much to validate everything that we theorize about experimentally. And the ideas from our group often start from an interesting problem in physics or engineering, which we then draft, solve, and find applications. So it's really a bottom-up application, and that's why I call this talk Transport Theory Guided Innovation in Microfluidics. This talk is an update of a talk that I gave at Nanobiotech Montreux in Switzerland last November, and I want to acknowledge the people who contributed significantly to this talk here. So as some of you know, hopefully there are a few people from microfluidics. Microfluidics is an evolving field. It's about, one can argue that maybe 40 years old, but there's been major development in the past 20 years in microfluidics. And it's been defined up until 2006, at least, by one of the pioneers and most influential figure in the field of microfluidics, Professor George Whitesides from Harvard. He describes in this very insightful Nature review that microfluidics is the manipulation in channels of fluids, in channels of dimensions of tens to hundreds of micrometers. And the idea is that microfluidics really emerged as a field. It's kind of a microplumbing in a way, where you inject fluids and you control fluids at the micro scale to perform several functions that you would do to just in a regular biology lab. And the idea is to accelerate, multiplex, and make cheaper the typical manipulations that you need to do in order to perform biological experiments. And so it's all about control of fluids in microchannels. For those of you who are microfluidicists, this is really obvious what I'm saying right now. However, in the past decade, maybe a decade and a half, microfluidics has branched into many, many different forms. So typically, as George Whitesides described, we think of microfluidics as channels, microcircuits of rectangular channels, which are used as mixers, diluters, to bring samples to detection zones, point of interest, etc. But there's been other ways to think about manipulating fluids at the micro scale. And one very important one I will be touching about in this talk is the concept of open space microfluidics. So instead of bringing fluids in channels, so in a planar chip, what we do instead is we bring fluids to an open surface. Say, imagine, for example, a petri dish, onto which we put a set of vertical openings, and we come and inject fluids very, very close to the bottom surface of the petri dish. So all of this is immersed. And the idea now is that we have kind of a shower-like approach to delivering fluids and reagents rather than a plumbing channel-like approach. And this has many advantages. Open microfluidics allows direct access to samples. You can interface it very easily with petri dishes. And it's compatible with large surface and automated liquid handling. And so in this talk, I'll be talking about what we call 1D microfluidics and 2D microfluidics. And here's an example of what I mean. So 1D microfluidics is a regular network of channels where you bring several fluids into, for example, here into a main channel. This is a classical experiment, which was the cover of Science, where you actually see that those fluids don't mix at the microscale, which is an interesting property to exploit. And then 2D microfluidics is where you inject and aspirate reagents from apertures located above a surface. And in that case, you have an unconfined flow. So you're not confined by channels, but rather by the fact that they're not mixing and the fluids are pushing onto each other and creating intricate patterns. And so there's a very strong analogy with electrical engineering here, where this would be treated as a network of resistances, and the pressure would be analogous to the voltage at each one of those branches, and the current would be analogous to the flow rate inside these channels. And here it's the same electrical analogy exists, which is called the electric hydraulic analogy, where now we're injecting current into a thin plate, and we're looking at the field lines of the flow, and that's why we call this field-based microfluidics. And so, first equation, we won't discuss solutions or anything, but for those of you who are engineers, it's nice to see where those phenomena come from. All those phenomena are governed, in both cases, are governed by convection, diffusion, and reaction at the microscale, which is described by this partial differential equation. The challenge in dealing with those 2D flow fields is that this velocity here, the velocity profile inside those devices is non-trivial, while it is rather trivial. It's linear in those branches. And so solving for this equation analytically is very, very hard for these systems. But very interesting. So, so far, no one had actually studied microfluidics in the context of flow fields and develop a theory so that we're able to do interesting design with these systems as of these systems. And so what I will talk today about is different applications of microfluidics, which range in either in the closed regime. So here's what I call the microfluidics openness matrix. So here we have networks of 1D channels on this line, 2D flow fields on this line, and there are many ways to use microfluidics in either closed or open format. And many researchers have done it. This is really the classical microfluidics here in this top left corner, but group from Seattle, for example, Ashley Taberge, or a group from Andras Hirleman in ETH Zurich and Basel campus. They've done open microfluidics in different channels, while other groups have used flow fields in microfluidics increasingly. And those are described here. And so during this talk, I will use this little openness matrix here to see where we are. And I will present 3 applications, one from each of these 3 squared. Classical one, open space microfluidics, and field-based microfluidics in closed channels, all with translational applications. So we'll start from the bottom. Here's the bottom, and now we'll go to the applications. So the first example I want to give of theory-guided innovation in microfluidics is to study the transport of nutrients in 3D biological samples. So as Professor Savoggi mentioned in his introduction, I'm also a researcher at the University of Montreal's Research Hospital, the CHUM, and member of the Institut du Cancer de Montréal, the Montreal Cancer Institute, and collaborate with cancer biologists and clinical oncologists on a regular basis. And one of the key issues that they're dealing with is to be able to analyze patient tumors and to be able to predict the outcome of a treatment before giving it to the patient, given there are a very large number of non-responders in cancer. So many chemotherapies do not work and provide only side effects for the patients. And so one key idea that was brought to me, and this I credit Dr. Anne-Marie Mesmasson from the CHUM for starting this, is this idea that if we could make primary tumors, that is pieces of tumors from patient, if we could make them survive on chip, we could use them to probe drugs and to verify whether the drug worked directly on the patient's tumor before prescribing it to the patient. So this is really opening this field of tissue explants and trying to use them as companion diagnostics. Alternatively, we could use those tissue explants to try to predict the efficacy of new drug candidates, which we have just simply tested on 2D cell lines. And if we can test them on primary tumors, we will have an extremely realistic model to see if the drugs actually can have therapeutic effects to cure cancer. But the first challenge, and this is something that we've been looking at for 10 years now, is how to make primary tumors survive on chip. And so the idea is in the body, tumors are, of course, they're perfused by the tumors, by the tumor vasculature, and they're supplied in oxygen and glucose and other nutrients, and then metabolites are evacuated. If we now cut tumors and grow them on chip, we no longer have access to that vasculature. And so the key idea is that if we cut tumors small enough, then passive diffusion of oxygen and glucose inside the tumor and passive diffusion of nutrients outside the tumor should compensate for the lack of vasculature. And so this is a problem that has been studied for the first time. We looked deep into the literature to find who has studied this first, and was done by Tomlinson and Gray, who studied in 1955, so a long time ago, the necrosis inside samples of lung tissue. And they realized that the tumors, if you cut them, if you excise them and grow them outside the body, would have only about 100 to 150 micron thick layer of viable tissue on the surface, and then all the rest would be necrotic. So we used a similar approach where we cut tissue and try to analyze it and simply calculate at first using diffusion reaction equation how small that tissue has to be to survive with passive diffusion. And if we do this calculation, we actually find that without perfusing the tissue, the maximum diameter to prevent necrosis, so prevent anoxia inside and consequently necrosis, is about 450 microns. And if we actually perfuse it, we can increase that to about 700 microns, but not much more. So this is a key intuition that got us into trying to slice tissue under these diameters to be able to make them survive. And so we tried that that was a very first in microfluidics, to microdissect tissue to sizes of about 400 microns. And we designed a chip, which is a very simple device with traps. So it has what we call these sedimentation traps here, where the tumors are heavier than water. And if you float them slowly above a trap, they'll just simply fall into it and remain there. And then you can change the medium easily in those chips later. And so with about 1 cubic centimeter of tumor, we can create a very large number of those micro-dissected tissue, or MDTs, load them into a large number of chips. Here we have chips that contain 8 samples per well. And we can also, so we can make one different experiment per set of 8 microdissected tissue. And so we can make 60 to 120 experimental conditions with 1 cubic centimeter of primary tumor. Before we go into the results and what we can achieve, we also had to develop a methodology to analyze those microdissected tissue. So the first intuitive approach was to use confocal microscopy. Unfortunately, optical methods are difficult to use with large 3D tissue samples, so sub-millimeter size, but still fairly large, because light doesn't penetrate in those tissues. So you only get an idea of what's happening at the surface of the tissue and never inside. Plus, it was difficult to convince clinical oncologists and pathologists that our results were relevant to their in translational science in general. So very early, we tried to create a chip and create a general process that is compatible with the gold standard in clinical oncology, which is to analyze tissue using histopathology and immunofluorescence. So we created this chip, and shortly after our first publication, where once we're done with an experiment, so we've exposed those tissue to drugs, for example, we've rinsed them, and before analysis, we insert formalin directly into those channels, we dehydrate the samples directly into the microfluidic chip, and we make it peelable such that we can peel off the top and be left just with a matrix of small tissue, which we then put in a paraffin embedding cassette. We cast liquid paraffin on it, peel off the paraffin, and remelt to have the samples align directly at the surface. And once we have that, we have a paraffin block, which contains, in this case, 32 microdissected tissue. And now when we take a slice of this block, we have access to several different samples, which we can look on the same cassette. So if for those of you who are familiar with tumor or tissue microarrays, TMAs, in pathology, there's basically blocks in which we put a core. So we drill a core from a patient block, and we insert it into a block, and we make an array of maybe 100 patients onto a single paraffin block, so that we can stain that block and look at a whole population of patients. Here we have something different but very similar, which we call the microdissected tissue microarray, so an MDTMA, where instead of having several patients, we have one patient, but every one of those lines here in the block, or those columns, is treated with a different drug. And so we can look at the patient's tissue treated with different reagents on a single block. So that's one of an achievement. Another thing we characterized is actually we needed to prove that those MDTs were better more viable than the current gold standard in tissue explants, which are tissue slices. So there's another student of our group, Dina Dorigiev, who's finishing her PhD right now, tackled this problem, where she compared. So she made tissue slices and microdissected tissue, looked at oxygen transported in, and our hypothesis was that the microdissected tissue would be much more viable than the tissue slices in a well plate or in a device. And that's what she could prove, actually, that this technology enables us to keep unperfused MDTs alive for over 15 days, while slices of the same thickness of roughly 300 to 400 micron thickness would not survive because of lack of access to sufficient nutrients. And so with these results, we started using our model to try to predict whether patients respond to treatment the same way that MDTs respond to the same treatment on chip. So basically, our ultimate goal is to be able, with these MDT chips, to predict what happened with patients. So we're not there yet, but we did start a preclinical study on 17 patients who donated samples of their tumor during the initial surgery, and then they were given an anti-cancer treatment. So those are ovarian cancer patients who were given carboplatin treatment. And so we gave the same treatment on chip to the devices, and we're actually analyzing the data right now. But we have, maybe next time I'll be able to tell you about our results, which I believe are quite promising at this point. But in the meantime, we also did those experiments on mice, where we used xenograft mice who developed tumors. And we could actually measure that depending on the cell line, whether they're carboplatin resistant or not. We could observe using our method that, for example, here, in OV1946, which is a carboplatin-sensitive cell line. So using primary mouse tumor tissue here, we can observe that we see a difference in cleave caspase 3, which means that the cells are more apoptotic when they're entering treatment in this case. Well, in the case of TOV112V, they're not. So this mouse patient here would be a responder, while this one here would be a non-responder. And the idea is to try to predict this before we give a treatment ultimately in patients. Another application of 3D biology on chip I want to touch on, and this is ongoing work in our group, is to use these microdissected tissue or any 3D biological samples to improve drug discovery, actually to ensure that what has been screened for in the early stage, so in phase 1 clinical trials or preclinical trials, is eventually is relevant for our good therapeutics. And we know that in biology, one of the challenges actually in pharmaceutical research is that screening on 2D cell lines can provide a lot of false positives in terms of therapeutic effects, because 2D cell lines are not they're not very predictive of what happens in 3D biology. So there's a growing consensus among cancer biologists that two-dimensional samples are less relevant than 3D biological samples for drug testing. And this, for this project, I'm working with a radio-oncologist, Dr. Philip Wong from the Princess Margaret Hospital in Toronto. And the idea of this project is, can we actually not only use drugs on our 3D samples, but can we also use radiotherapy? And so the idea is to generate a protocol by which we will, instead of testing patients, we will test a large number of spheroids or microdissected tissue onto a chip and treat them as patients in clinical grade radiotherapy machines. And so the idea is that with such an approach, and this is really an open problem, and if anyone has interest in 3D biology, I encourage you to think about the applications also in radiotherapy. The idea is that, as you may know, the pharmaceutical companies are sitting on hundreds of thousands, sometimes millions of compounds they have developed, but who did not go through clinical trials or who failed early clinical trials. And many of these compounds were never tested with radiotherapy. And radiotherapy only comes extremely late in the validation process, typically in a clinical phase 3B study. So after they've been approved in phase 3. And if we can bring radiotherapy, if we can find a way to have a moderate to high throughput to bring radiotherapy early on in drug discovery, well, maybe some of these compounds, which would have synergistic effects with chemotherapy, and maybe some of the compounds they develop would become now interesting candidates to go through the clinical trial pipeline. And so if we can save a fraction of 1% of compounds and give them a second chance, we're basically creating this compound recycling program for the pharma industry. And that's what we call the SMARTS, the Search for Molecular Agent Radiotherapy Synergies program, which is ongoing in our groups. And so several students have started looking at this, and we started this project with spheroids this time, which are simplified tumors, right? They're just aggregates of cells. And again, we've shown using models and diffusion reaction models that we can make very large spheroids that would exhibit naturally hypoxia, as we see in tissue. So those spheroids are bigger than the microdissected tissue, as I've shown. They're about 800 microns in size. And if we make them this way, we have about a 50% surface area here that is normoxic, that has regular access to oxygen, and about 50% of the area which is hypoxic, which has very low oxygen. And then when we test drugs or radiotherapy on these systems, we can immediately see the effect of oxygen in these on the compounds or treatments. And so we've started this this program looking at various combinations of radiotherapy and chemotherapy, and we have published actually 3 papers on this topic so far. But the idea is that in many cases we can we perform tests with different drug concentration and also with different radiotherapy doses here, 0 gray, 2 gray, and 4 gray. And the idea is that in cases where the product of the survival fraction from both radiotherapy and chemotherapy is less than the product of these, or in other words, when log of A here plus log of B here is larger than log of C, then we have a synergy, which means that together those kill more than they would do sequentially if they were only additive. And so with this framework, we could validate that several agents, especially those affecting DNA repair, so those inhibiting DNA repair, like PARP inhibitors, are actually very synergistic with radiotherapy, and that creates an avenue for trying to integrate these treatments early on. And we wrote recently a review which you can refer to that basically describes our work, but especially other groups' work in terms of using radiobiology and radiation therapy on 3D biological samples. So you can read this. It was published about 2 months ago in LabChip. So I want to conclude this chapter 1 with just the idea that all the concept of microdissected tumor on chip has been now transformed into a spin-off from my group and Professor Philip Wong's group in Toronto. And we're basically, so the spin-off is called MesoChip. So this is a brief presentation to show that MesoChip exists, that we're actually raising funds now, we're recruiting as well. We have 3 full-time employees. And it's also a disclaimer that I have interest in this company and in the commercialization of microdissected tissue. So if you want more information about MesoChip, you can check online and contact our CEO, Jean-Luc, or myself, and be happy to tell you more about it. So now if we move to chapter 2 of this talk, I'd like now to show another example of theory-guided innovation in microfluidics, which is how to use what we call multipolar microfluidic systems for biological surface processing. So the last 2 chapters will be are more early stage. We haven't reached yet to the point where we're using it with patient tissue, but it's coming for chapter 2. So we'll go back to engineering a little bit. And the goal of this project now is to be able actually to study how we can understand, first of all, how when we put several point sources, so when we inject reagents above a surface, how we can create these intricate patterns. There's been this paper published, so the middle panel here describes here an experiment done by David Juncker, and so in Nature Materials, which gave birth to the field of microfluidic probes, which are used in several now are used in several groups for surface processing. But the idea, the first part, we're physicists at the core, and so the first part was develop methods and new ways to see microfluidics so that we can model these intricate patterns. And so I will skip much of the theory, which was developed by Etienne Boulet, who did an absolute absolutely great job on this. He's finishing his PhD right now in my group. Just to mention that one very important property of 2D flow field microfluidic systems is that they can be described as flow in thin gaps, which are called Healy-Shaw flows in fluid dynamics. And that whenever we have an experiment where we can actually we can observe one of these flows, then we can use undergraduate level mathematics, so complex variable analysis, to transform an image into another one. So we can using a complex function, and in doing so, we can scan and try to find other patterns that we can create experimentally. And this is what Etienne Boulet has done first theoretically. So he's shown that by using complex transform, you can transform an experimental image into another one, which you can create later. And the reason why there's a chessboard here is that the analogy is that if you take this chessboard here and square it, so you take a complex variable and transform it into a different board, the board is completely different, but the rules of chess would be preserved on this second board here. And it's the same here in this very exotic one, which is hard to imagine. But the idea is by transforming an image using what we call a conformal transform, you transform the image, but the physics remain intact. The laws of fluid flow and transport remains intact. And that is what we use. This is really the basic mathematical concept that we use to create what we call multipolar microfluidic devices. And those are really real and were built and tested. So the way they work, here you have a 3D printed device, which is basically a nozzle with many openings, and each of these openings is linked to a tube and ultimately to a manifold and to a pump. And then by either pushing fluids or aspirating it through these channels, you can create intricate patterns on a surface. So you bring this probe to a surface, and then you, for example, if you aspirate here you make a flower configuration, you aspirate at the center and you inject different reagents on the side, which are varying in time, you can make patterns of reagents like this one appearing on a surface. Okay, so now you can control reagents on a surface, and you can have this flicker and alternate to treat parts of a petri dish with different reagents, and by modulating the frequency and the spatial resolution in real time. So this is a tool to basically process surfaces that we have developed. And the first thing that we did in terms of translating this to the life sciences is to automate a basic ELISA immunoassay. Actually, it's a sandwich fluorescent assay here that we built. So we brought this 12 aperture system above a glass slide, which is patterned with dots containing capture antibodies. And this is an experiment that was done in collaboration with David Younker's group at McGill, who provided the immuno glass slide here. And then we injected with the device 6 different concentrations of an antigen. We wanted to capture one of each in these different the petals of this flower. Then after a certain time, we stopped injecting. We injected the detection antibodies for another, incubated for another 50 minutes. And then we rinsed the surface with PBST with a final aperture. And once we were done, we could just dry the slide and image it. And here we see on the slide, so the different concentrations of antigens bound on different parts and making this sort of flower here. And so we could create an entirely automated immunoassay with limits of detection comparable to what is done in regular assays. And so there's potential for this technology to automate different assays this way. And so Dr. Pierre-Alexandre Goyette, who's a former PhD student in my group, just graduated, tried to develop very large systems, which are now, instead of being sort of a flower-like system, we tried to develop a pixelized screen that could project reagents onto a surface. So we developed these large arrays, and the manifolds required to be able to bring what we call a pixelated chemical display. So this is now a nozzle with many, many holes that we bring very close to a surface, and then we stream reagents. We inject reagents from some apertures, and we aspirate them back from other nearby apertures. And in doing so, we can confine flow to very clear-cut pixels that we can then modulate. And so Pierre-Alexandre, oh, excuse me, this. Excuse me here. I will stop this. Okay, I will just skip. So that's Pierre-Alexandre presenting. I don't know why the sound is coming. It's supposed to be off. But the idea is that we created this device here, which is basically, as you see, is just dropped onto a Petri dish. So this is our PCD. It's dropped there, and then we can perform an experiment. And so we've tried to show as a proof of principle that we can use this to pattern plastic films in a roll-to-roll process where we just bring the PCD to the surface, and we can make these pattern of fluorescent proteins adsorbed to the film. And we also demonstrated we can use interfacial reactions between certain of the pixel, for example, to deposit here silver wire directly on the plastic film. So we made some of these proof of concept for surface processing, and the ultimate goal was to create the first microfluidic movie, so pixelated movie here, where we actually use a manifold and valves to control the reagents in every one of these pixels, actually here in groups of pixels. So you have to think of these old liquid crystal watches where you could have a line of pixels appear at a certain time. So that's back in the 80s and 90s. That's where we are in terms of controlling our pixels. But here's a result here of a movie where we can actually control the content of pixel in terms of reagent, and our first movie spells Poly for the name of our institution, Polytechnique Montréal. And so we've shown that we can create high-resolution surface patterns with these systems and also control them in time to deliver different reagents in an automated way to a surface. So it's 12:50. We started a bit late, but I have maybe 10 more minutes to discuss chapter 3, and then I'll be happy to stay as long as you guys want for questions. But this last chapter here is again a bit different, but it's also theory-guided innovation. And now this is happening in capillary microfluidics. So in microfluidic circuits that are propelled by capillary forces. And what I will discuss is the investigation of the phenomena of self-coalescence, and this time for point-of-care testing and high-throughput screening in microfluidic systems. So as you may know, you certainly know if you're in translational research, most of pharmaceutical research is performed currently with robotic liquid handlers. And this is what Professor Steve Quake from Stanford aptly calls the tyranny of pipetting. And he's presented a talk at the Nobel Symposium in 2017 called Solving the Tyranny of Pipetting using Microfluidics, which you can access on arXiv. It's very interesting overview of the field of microfluidics and how it has evolved. So basically, microfluidics is there to make those robotic actions more efficient, right? Because even though today we have very effective robotic liquid handlers, if you look carefully, they are a mere robotization of the human pipetting movement, except that you have one arm carrying several pipettes at the same time. There's definitely room for improvement in terms of delivering reagents into small volumes. Plus, the volumes of 384 well plates or even 1,536 well plates are still fairly large for microfluidic standards, which means that you have about 5 microliter or 5 cubic millimeters of fluid per well in a 1536 well plate. Using microfluidic systems, you can make compartments that are less than 1 nanoliter in size. And this was also demonstrated early by Steve Quake. And so in this last project, our goal is to use capillary microfluidics to create microcompartments containing very small amount of reagents in high concentration to perform high throughput screening. But we will go through this in a very sequential manner, starting from experimental observations. One of the key challenges in microfluidics since the very beginning of the field is that we can make is how to integrate reagents in microchannels. We can make channels very precisely and control fluids in very precise manner, but when we integrate reagents, something always happens. And here's a channel, for example, in which we have, this is a 500 micron channel wide, 500 micron wide channel here, into which we have printed reagents, simply a food coloring here that dried, and now we introduce water into this channel. And here's what happens. So as soon as the water touches the dried reagents, it gets reconstituted, so rehydrated. It appears in solution, but one key issue here that prevents microfluidic systems from handling complex, sequentially complex chemical reactions is that all those reagents agglomerate at the air-water interface at the very end. So they're completely mixed together, and if you had several different reagents here in the end, they would all be mixed together. That can be useful in some applications, but in most applications, it's a drawback. So this work here that I describe is a collaboration with IBM Research Europe, the Zurich laboratory in Switzerland, where I had the chance to collaborate with Professor Emmanuel de la Marche's team since 2014, and where I actually I was hired for 1 year in 2019 to work with them over in Europe. So we'll start this video again, where the team at IBM had discovered a very interesting solution to excuse me, this is starting, okay, the play. They found a very interesting solution to prevent reagents from being dispersed and accumulated in a single spot. And this is called self-coalescence, actually. And the way that this happens is, I will show it first. So here's the result. You introduce a flow, and in this case, there's absolutely, excuse me again, the video is finicky. So once you've finished resuspending the reagents, they are fully, they preserve their original orientation and then can be sent to another module here for analysis or detection. So the key to this effect here is what I will stop the video. Is what we call it's capillary self-coalescence. So the idea is to bring a fluid along what we call a capillary pinning line. Here we have etched a 5 micron deep and 5 micron wide trench into silicon. And when the fluid comes, this trench here surprisingly acts like a capillary barrier, which prevents the fluid from spilling over in the second part. And then when this channel fills, then the fluid turns, and when it turns at the very end here and comes back, it touches its own air-water interface. And so the part on the right here coalesces with the part on the left. And in doing so, the fluid crosses this trench perpendicularly rather than axially. And this is really what creates this this non-dispersive effect on reagent dots. And if you look at this using a particle image velocimetry, for example, you see that so when the particles, so the streak lines give you an idea of the velocity of the particle, and you see that velocity of the particle is maximum near the air-water interface. But as soon as the interface goes, those particles become simple dots, which means they stopped. So they become stagnant. So in this situation, we have here a flow that is concentrated only near the air-water interface. And so we call this zipper flow physics because it's similar to a mechanical zipper in case where the wave propagates with the slider here, so laterally. But what you do is you actually bring the teeth of the zipper perpendicularly to the direction of the slider, and as soon as the slider has passed, those teeth are stationary behind. So that's why we also call it zipper flow physics. And we've done some modeling using conformal mapping and trying to understand how this happens, and we could get a very accurate model of this effect using the techniques that Etienne Boulet developed in the previous chapter. So using 2D flow field physics happening this time in microchannels. And we could find that this decay in velocity is exponential. And so this could give us a very good idea of why the samples remain on site and do not disperse during reconstitution using self-coalescence. And this was the work done by Samuel Castonguay, also finishing his PhD in my group right now. And so we've now used these devices. I will go briefly over the applications, but we've used these devices for, first of all, looking at viral detection. So we used the HPV virus, which we tried to detect. So the idea was to do a PCR inside the microfluidic chip to detect a virus. So we prepared the isothermal PCR mixture, and so which is here. We spotted this using a piezoelectric droplet dispenser into our channel. We then sealed our channel and we brought in the virus sample, which got into the channel. DNA got amplified, and we could measure the presence of the virus. So this is in a way, well, this was published in 2019. If we had published this 2-3 years later, we would have certainly detected coronavirus with this system. But the idea is that this type of microsystem could eventually lead to the creation of rapid diagnostic tests like those that you've taken at home for the past 2 years on a regular basis. If you're like me and you travel a lot, I think I'm up to about 20 or 21 negative tests for COVID since the beginning of the pandemic. But the idea is that so far we only had at home, we only have the antigenic test, so the immunoassay test using a lateral flow strip. Why? Because this is extremely simple to do. We don't have portable PCR tests at home, and we still have to go to the hospital or the clinic to have this done. So what we propose here is a general architecture in which, by simply inserting a sample into a device, we could get a portable PCR testing system at home. And so that's what IBM is working on. And so this is one test that we've done. Then we also measured, tried to identify enzymatic deficiencies in blood using these devices by spotting different reagents and bringing in an enzyme and looking at the intensity of how this enzyme converts the substrates into fluorescent or colorful material, and you could achieve enzymatic activity assays in this system. And I want to take 2-3 more minutes to show how this could be brought to high throughput systems. So this is my work while I was hosted at IBM Research in Zurich. We tried to make these self-coalescence module in a multiplex fashion. So here's a device where there are 15 parallel self-coalescence modules here. Each are spotted with reagent patterns. Then the devices are sealed with a dry film. Then we just add a very small amount of water and watch it fill, because it all fills using capillary action. So here's the device in action here. So you will see the same self-coalescence effect, except now that we have 15 systems in parallel, and this is entirely propelled by capillary action. So there are no pumps, nothing used to create this this flow effect. So reagents get reconstituted, and they can interact with whatever is in the liquid sample we have brought in. So we've done this with, we've shown that we can do this with different reagent patterns. So this is the same thing here using an aesthetic design. So we looked at the rose in a jar from the Little Prince from Saint-Exupéry, and that we were inspired by this. And so here, same thing, you can reconstitute several reagents, but now we use here fluorescent dextrans just to show where the reagents are and where they stay. And what we observe is once we've reconstituted them, so we can image them using simply a fluorescence microscope. And then if we wait long enough, those reagents diffuse and start to mix together. But it takes quite a bit of time before they diffuse one into the other if they are adequately spaced on the chip. And so we've done an analysis of how these systems can how reagents interact via diffusion in those channels. So again, back to reaction diffusion theory here to show, and we developed basically guidelines to say that if you don't want your reagents to mix in an experiment of, say, 8 minutes here, then you have to space them in a certain way. And if you do want them to mix, then they have to be within a certain distance of each other to mix during the time. So what we're doing with this is we're either forcing mixing of two reagents or preventing mixing of two reagents by using simply distance of spotting between them. So we can make mixing or what we call virtual compartmentalization of these reagents. And so models were very useful to create the experiments, and the experiments yielded fairly similar results to the models. And what we want to do with this now is to, we want to show that we have an architecture in which we can store a lot of chemical data or of a lot of different chemicals in either in a way such that they can mix together or remain separated, and use this to perform high throughput experiments. And that's where we are right now. And this is our ultimate feat, which is 100 self-coalescence modules in parallel, each containing 100 dots of reagents in a 3-bit format. So the concentrations could be anywhere from 0 to 8x. And we could create this universally recognizable work of art using our system. So we do have precise control over the spotting and reconstitution of those reagents in a multiplex fashion. And I want to acknowledge especially the work of Dr. Yüksel Temiz and Dr. Emmanuel Delamarche, who actually no longer are at IBM right now. They went on to different projects, but this was a very exciting project, and it's still an ongoing project in our group. So with all this, I'd like to conclude, and thank you for your patience. My conclusion is that to make good microfluidic design, you need good models. And microfluidic is now a multi-dimensional field. It's not just about flowing liquids and channels. It can be much more complex than this. And many more new fundamental microfluidic applications remain to be discovered in the 2D flow field application, which format which we propose here. And there are also other exciting applications in automated liquid handling, point-of-care testing, and tissue on chip for both open and closed microfluidics. So if you're interested in microfluidics, I can say that also I'm the scientific co-director of the microfluidics core facility at the University of Montreal's Research Hospital, and we offer a lot of technologies. Actually, if you want to try them, we can help you. We can provide you, especially for the tissue on chip part. And if you want to discuss the other applications, well, you have my my contact now, and I just want to end up by acknowledging the work of very talented team of students, many of you whom here have participated in this work. So thanks a lot for your attention, and I'll be happy to answer any questions.
Well, thank you very much, Professor Gervais, for your excellent presentation. Very nice results, and I truly enjoyed it. So I encourage the participants to ask their questions using the Ask a Question box. I see that like we see some communications with our speaker in the chat box as well. Thomas, it seems that people are enjoying. They enjoyed your talk, and it's a great actually feedback that you received for this presentation. Do we have a question in the Ask a Question box?
Just before that, Tuma can stop sharing. You know, there is a cross if you go with your mouse in the middle of your, first. Thank you.
Awesome. So Marco has a question from you in the Ask a Question box. I'm much enjoying your talk. Could you give advantages of multipolar printheads over microarray inkjets? So they want to know what is the difference between these, open concept printheads that you have developed.
Excuse me, the communication is a bit choppy. I have missed the difference between what exactly?
So the printheads that you developed in chapter 2, I believe, over
inkjets, inkjet printer. Yeah.
Inkjets, yes.
So
yes.
The idea of if you are to use an inkjet printer, so you could print on dry surfaces. You can also print immersed, provided your reagents are heavier than water, and this has been demonstrated, for example, by the Takayama group. The main advantage of our technique is that you can shift the reagent in time. So basically, you can create a pattern and then either move the surface below, like in a roll-to-roll system, or change the pattern, as we've shown, to project different reagents in time. And when you project a new reagent, you completely remove the previous reagent. Whereas if you print, if you do spotting, you would what would happen is that whatever you spotted would diffuse everywhere into the system. So by using continuous streaming of reagents, you can confine spatially the region. While if you just spot on an open surface, everything will mix up around. This, of course, has a drawback, which is that in order to do continuous streaming, you have to spend a lot of fluid doing things. So if you're using very expensive reagents, those pixelated chemical displays are less useful, and because you can find another way to print reagents. But if you use cheap reagents such as small molecule chemotherapy drugs, fluorescent markers, etc., they're quite convenient and higher resolution.
Wonderful. So Marco, I hope you got the answer that you're looking for. So my question is, Thomas, is primarily about, most of my question about the first chapter that you talked about, this microfluidic device for testing drugs on tissue biopsies. So my first question is about the material that you use to create these microfluidic devices. I assume that you use PDMS. Is that correct?
In most cases, yes. We have other, we're actually investigating right now other thermoplastic materials that could be used to do this, but that are still porous enough to let oxygen through. I mean, PDMS is not liked in the pharmaceutical research because it absorbs small molecules. However, we still have to see another polymer which is as porous to oxygen as the thermoset silicon-based PDMS. And I mean, it's so porous to oxygen, and when you think of growing cells in PDMS samples, you can imagine removing the PDMS and have like the fluid channel float in air, and that's basically the access you have to oxygen. There's no barrier by the PDMS, who's about 50 times more permeable than water itself.
I see.
So we have to find, it's not perfect, but if we don't use PDMS, we have to think of much more complex strategies to bring the oxygen to the site.
It's very nice. You answered my question before I even asked it. So, but then I have another question about PDMS in terms of radiation therapy. Did you notice any challenges with PDMS interacting with the radiation doses that you use in terms of releasing toxic elements or compounds that can be can affect?
So I'm not a medical physicist, but we did watch this effect closely, and what I can say is that the modeling system that modeled dose delivery in patients are extremely well developed and precise, and they use this concept of atom density and water-like equivalence. So if you bring a new material, you can model it and compare it in terms of water-like equivalence. So how many times is it more absorbent to the dose than water? And what we find is that PDMS has a water equivalence of approximately 1, which means it will just behave like a layer of water in terms of radiotherapy dose absorption. And it can be handled very readily by the current treatment planning softwares in radiotherapy clinics.
Okay.
So there's no problem there. It's actually a good news that it's a highly compatible material.
That's great. That's great. So I have one more question, primarily around the translation of these technologies that you developed. It's a broad question. What are the challenges of translating these technologies into clinic? I mean, you can pick any of the technologies that you developed, and then maybe you can.
So, yeah, 1 question, 3 answers, 1 per chapter that I presented. For chapter 1, which is 3D biology on chip, I would say that the main challenge is to, the very first challenge is to prove that those microdissected tissue are indeed a good proxy for human patients, right? The intuition is there. I mean, we go from 2D cell culture to spheroids, which are avascular cell aggregates without any extracellular matrix, to something that is a piece of tissue which has several cell lines interacting together in an extracellular matrix with remnants of vasculature. It's a real tissue. So the common sense would tell us that this will recapitulate what happens in the human body. But at what cost, right? They're more difficult to use than the spheroids. So what we are trying hard to do is to prove that the extra effort in growing them and maintaining them, because they're more difficult to culture than spheroids, is worth the realism that they bring. And we have to convince clinicians and the pharmaceutical industry that this is the case. Secondly, we need to convince them that the chip-based format is also worth the extra effort, because in the pharma industry, everything is done using robotic liquid handlers. So our tumors, our MDTs survive way better in a PDMS vessel than in wells, for the reason I mentioned about oxygen transport. So we need to work to convince them with our research that they are indeed worth implementing in high throughput laboratories in the pharma industry. So those are the main challenges for chapter 1. The main challenge for chapter 2, I mean, the chapter 2 is more early stage, so I think we still have to work on the engineering architecture of the system to make them really versatile. And we also need to find a killer application. So what we're currently working on with these pixelated chemical display, the first killer application we're currently investigating is to use them to perform dynamic or frequency modulated stimulation of cell patterns to probe signaling pathways. So there's an emerging consensus that signaling pathways are not only a matter of how much drug you put, but at what frequency you put those drugs. And there's been evidence that if you give a drug to a cell in a continuous fashion or in a pulse fashion, you can induce opposite phenotypes like survival or death. And this is really something that we're not using in drug development, this concept of dynamic control of drug delivery. So we're using these devices as a way to multiplex the testing of drugs. But it's more early stage, and we have to find applications where reagents are cheap, as I said, because we use a large fraction compared to robotic liquid handlers. And if we have good results on this, some people will follow up and want to try it. And for the last one, I would say the challenge is really in order to, say, bring those capillary device to your house and make them available in the next pandemic so that we can have PCR tests at home and in airports in less than 30 minutes. It's really it's a feat of control. I mean, and it's a lot about creating devices that work reproducibly in uncontrolled environments where temperature can vary, and this affects capillary action, and handling complexity. And so we could do this in a laboratory under very controlled conditions, but there are still some steps to do to prove that you can just distribute it. But IBM research at this moment has everything needed to fabricate those devices. The device architecture is there working, it's robust. It's now how to handle the complexity of testing. But this is something we're currently investigating with other groups in the world. So awesome. Here are my 3 answers for very
comprehensive. Really, I love that you touched it. I gave you an option to just go with like one, but you provided like answers to all of these. Very, very comprehensive. So I don't have more questions, but Hooman, if you have any, feel free to ask.
No, first of all, I want to thank Professor Gervais for this great talk. I enjoyed every minute of your speech, and I learned a lot. Hopefully, there is no future pandemic, so we can find another application for your PCR chip.
Hopefully, but I wouldn't count on it.
Other than pandemic.
There's multipoles coming, I think, so.
So yeah, I just want to end this session by thanking all the audience who participated in this talk, and also Professor Gervais, and also encourage everyone to join us next week at the same time for Professor Kathleen Granfield talk from McMaster University. So, and take care of yourself, and have a great
Take care.
day.
Thank you very much.
Thank you, everyone.
Thank you so much.
Bye.